All right, welcome back. This is Unit 2, Essay 1. So we're going to be talking about the muscles. Specifically, first we're going to be talking about skeletal muscle contraction. And that deals with the sarcomere. So, sarcomeres, just like draw this here. Okay. We got a couple things on here. So this is a Z disc. We've also got Titan. Pretty sure that's how you spell it. And then let's see what else we got here. We got we got the Z disc. So this is the sarcomere. And then we also have a couple other things on here. We've got F actin. This is G actin. So this G actin has like little binding sites. Oh boy, it doesn't look very good, but that is the G actin. Okay. So you also have tropomyosin. I'll put this in blue. So tropomyosin binds to the G actin. So then it kind of looks like this. Oh. We got some. Okay. And then we've got a couple different types of troponin. And this troponin kind of looks like a little Mickey Mouse head. So we got troponin T, troponin I, and troponin C. So troponin I is B-actin. Troponin T is tropomyosin. Troponin C is calcium. And then these are the myosin binding sites. Let's see, what else is there for the sarcomere? Let me think here. So there's thick filaments, which are made of myosin. And then the thin filaments. So here's myosin here. We'll put those. Not the best at drawing, but okay. So this is a myosin. So this binds to the actin, just over here. These are just like little, I don't even know how to draw them. Just kind of like that, just so that those could fit in there. And this little spot is called nebulin. Okay. So the myosin binds to the troponin. Well, these troponins bind here. And that will activate like a contraction. Let's see. Okay, so we also need to talk about the action potential and the calcium releasage. I don't know if releasage is the right word. But, so we've got the cell here. This is called the T-tubule. This is called the SR, but it's also called this. It's not the smoothie R, the sarcoplasmic reticulum is what it's called. And so there's a couple things that go on to here. So this is called the RYR. Come on. And then I'm trying to think. What else we have? Might need to check back here really quick. Okay, yeah, so the RIR and the DHP, that's what it is. DHP. Sorry about that. So the RIR and the DHP are closed. So we'll draw them a little closer. But, kind of like in here, they need to be opened by this, the calsequester. So the calsequester opens this channel so that the calcium can go into the ICF. So that is what is important with muscles. It's really important that the calcium gets into the ICF. Here's a little bit better of a picture. I can kind of explain this a little better as well. So we've got the SR right here. And so RIR and DHP are important with this. And you also have a synapse here where you have some sodium being allowed in the cell and potassium leaving the cell, kind of like normal. And that causes the calcium to go inside the cell as well. There is a lot of calcium inside the sarcoplasmic reticulum. No, sarcoplasmic reticulum, I think. I keep forgetting that word. And then, so you have these T tubules. They're kind of like, I don't know how I would compare them to something. I guess they activate these voltage gated channels. Just when, so the calcium is released through voltage gated channel, and then it will go down through here. Well, it'll also go through the RYR. Let me just draw this all out here. Okay, let me back up. I'm getting a little mixed up in my words here. So, the depolarization spreads down through the T tubules, which activates the voltage gated channels. And then the calcium binds to the troponin, which causes a shift of the protein, a conformational shift, which exposes the binding sites. Then the myosin heads, wow, I spelled myosin wrong over here. So the myosin heads then pull the actin toward the M line, which is like the power stroke. And I can draw the cycle as well. So let's draw the cycle here. My pen is not working here. I don't know why. Okay. So first, here's the Z line. Here's the actin. Okay, so we got here. This is called the cocked position. And then we also got a phosphate on there. So this cross bridge cycle uses ADP first. So then calcium is brought in. And then you got heat also leaves a little bit. So once calcium is brought in, this binds to the actin. It's still cocked. And the phosphate is still on here. But in this step, the phosphate actually leaves. So put that there, phosphate. And so the phosphate is stopping this from contracting. So ADP also leaves. And then this is called the power stroke. So this is called an uncocked position. And this is like the contraction that we're looking for here. Then ATP gets brought in. And then this is unbound. and still uncocked. And then this part is called hydrolysis. Okay. So this is for skeletal muscle. So we can talk a little bit about smooth muscle now, since we already have drawn this. Here, I'll copy this. And it's pretty similar. So there's two different kinds of this cycle for smooth muscle. So here, smooth. And there's a couple of different things that like disrupt this cycle, like MLCP, for example. So now we'll use blue to show the differences. So MLCK comes in as well as ATP. And then there is another phosphate on here. So this phosphate still leaves. And ADP also leaves still. That there. Okay. And so the power stroke, so this blue phosphate still stays on there. And then when it's uncocked, you still have this phosphate. So then this MLCP comes into play. So MLCP takes off the phosphate. And so then it'll bring you back to step one. So this is when MLCP comes in late. So we're going to draw the same thing. I guess I already copied it. Oh, not that. I'll just go like this. Copy. Okay. Okay. So this one is pretty similar, but the MLCP comes earlier. So that means that phosphate comes off earlier. So we can take it off over here. Okay, so you still have the MLCK and the ATP here. Because for the smooth muscle, you need that ATP. Smooth muscle uses ATP. So you got this phosphate on there, but we'll say that the MLCP comes in earlier. So that means the phosphate leaves. So then the power stroke still happens, and ATP goes in, but it also leaves. And then this is uncocked, and then it latches. So the phosphate is bound on there again. And this is like, so smooth muscle can stay contracted for a long time. So that's what this is kind of showing, is that this stays contracted. Let's see what else I have to talk about here. So, for muscle contraction, the energy source is ATP. And that lasts about 2 seconds, and it's pretty quick energy. Then you also have the phosphocreatine system, which is about 10 to 15 seconds. And that is like short bursts. Then you have glycolysis, which is about 1 minute. And then you have aerobic respiration, which is hours. So the first three are anaerobic, and I guess I should probably write this. So you got cytosolic ATP, creatine, phosphate, so that says 10 seconds. Glycolysis. Sorry, my handwriting is horrible right now. About a minute. And then you've got aerobic. I don't know how to spell that. I think that. So that takes about hours. So let's see. There's so fatigue is also kind of in this as well. So you've got physiological fatigue or psychological fatigue. So with the physiological fatigue, you've got low frequency and high frequency. So so low and high. So the high frequency is like just buildup of potassium in the tubule. That can be fixed by just voltage gated channels. Low frequencies buildup of magnesium, I'm pretty sure. Let's see what else here. I guess we can talk some more about smooth muscle. So skeletal muscle is voluntary, smooth muscle is not voluntary. So smooth muscle doesn't have sarcomeres, as well as it contracts slower, but like stays contracted for longer. Let's see. Yeah, that's pretty much all that I can think of with the smooth muscle. Let me think if there's anything else that I need to talk about for the prompt here. Let's see if Number 2, the essay. Include relative events. 2 main types of smooth muscle. Oh, so I guess I forgot to mention this earlier. So this is phasic. That's when the MLCP comes in late. This is tonic. So these are the types of smooth muscle. Okay, so with the muscle sensory receptors, there are muscle spindles, which kind of detect the stretch, and they initiate contraction so that you don't overstretch. Then you also have the Golgi somethings, which detect tension and inhibit contraction. Let's see. I think I needed to also talk about reflex. Let me see here. Oh, okay. So, this is G protein. So this is the binding site, and this G protein can have a conformational shift, and then phospholipase C is activated. Then you have all these different types of channels here. So you got voltage calcium channel, you've got stretch activated calcium channel, ligand gated channel, calcium channel, and calcium induced channel. So calcium is very, very important with the muscles. Okay, so with reflexes, you have most of the reflexes are involuntary, like it says right here. You've got motor neurons. You also have gamma motor neurons, as well as sensory fibers. So all of these are used to help you with your reflexes. You also have tendons as well. So the what is this called? The starts in the dorsal root ganglia. No, this is the ventral root ganglia. The ventral root. And then it leaves, and there's an agonist or an antagonist with these muscles. Like bicep, for example, is the agonist, and then the tricep works against that, and it is the antagonist. So yes, this is the ventral horn. Then so this EPSP, which is excitatory, and the IPSP is inhibitory. So the tendons do kind of similar things, where it sends like a reflex through these, I can't remember what the sensory fibers. But let me see here. What else I need to explain here? So yeah, for the smooth muscle contraction, calcium enters through just voltage gated channels, or this sarcoplasmic reticulum. I think I'm saying it right. I can't remember what the name word is. Releases that calcium. Let's see. Yeah. So yeah, we talked about just the organization of the muscle and contraction. And I know that I need to work on this a little bit more. I don't quite have it down all the way, so hopefully you have some mercy on me. But thank you for watching. See you next time.